Okay, we are gonna get started. 10 clockwise. This is not working. I need to mute this. Just 1 second, sorry. Forward. Okay, just as a reminder, there's a quiz today on Moodle. It's open till 11:59 p.m. So plan your day so that you're starting before 11:48. Your next test, also a reminder, is Wednesday the 2nd. It's gonna be on chapter 6 through 8. And homework 7 is due on Monday. It's a different format than we've done before. I'm trying to mix it up so you can think about what is good for you and what's not good for you. For some of you, maybe it's not gonna be that format. Some of you might love it, I don't know. It's just a chapter outline. So there's a template posted on Moodle, and you're gonna be filling in details as you're reading. So filling in those details as you read, you can add diagrams, whatever you want. You can print it and handwrite it, or you can type it. You know that I think typing is better, but I mean, handwriting is better than typing, but it's up to you. Okay, for some of you, typing is better than handwriting. No offense, I have bad handwriting too, as you might have seen. We need to move quickly. I was hoping to get through chapter 6 today. I'm not sure. We'll see what happens. But I've been meaning to share this verse all week. From Psalm 37. Psalm 37:23, this is the New Living Translation, says that the Lord directs the steps of the godly. He delights in every detail of their lives. As we're thinking about the details of the cell, I hope it's reassuring or comforting to you that God cares about the details of your life. I know at the end of the 4th week of the semester, you're tired, starting to feel more real in a lot of ways. So just remembering that God is delighting in this detail of your life. He's delighting in you learning about the details of his creation and of cells. Even if you're not delighting in it, he's delighting. So, as I said, I'm hoping to finish chapter 6 today. I went through last night, I went through my notes, and I highlighted things to try to keep myself from talking about every detail of every organelle so that we can maybe make progress a little bit faster. We'll see. Okay, so we finished the endomembrane system on Wednesday. And we are now working our way through the rest of the organelles, and then we're going to talk about how cells interact with each other to finish out chapter 6. So some of these ones that we're going to talk about today, I think you're probably a little more familiar with. Mitochondria is one that we talk about all the time. The next 2 organelles that we're going to talk about, their job is changing energy from one form to another. Okay, so mitochondria, when we talk about mitochondria, we always talk about the mitochondria being the powerhouse of the cell because they're making energy. I want you to think about that making of energy. We're talking about the reaction of cellular respiration, which again, we're going to come back to in detail in chapter 9. Cellular respiration is using oxygen and carbohydrates or fats or proteins to make ATP, which is adenosine triphosphate. ATP is the fuel for cells. So we're converting energy in the form of stored chemical bonds in carbohydrates or proteins or lipids, and we're adding oxygen, and we're changing that into a form of energy that your cells can use, the gas for your cells, which is ATP. And again, we'll be talking more about this in chapter 9 in more detail again than maybe you want to be talking about it. But all this is happening in the mitochondria. So mitochondria are found in plant cells. When we talk about chloroplasts next, everybody knows that chloroplasts are found in plant cells. Plant cells also have mitochondria. They still need energy. The benefit they have is they can make their own carbohydrates to fuel their mitochondria. So they're found in plants, they're found in fungi, they're found in animals, they're found in protists. They are not found in prokaryotes. Why are they not in prokaryotic cells? Because they're membrane bound, and prokaryotic cells don't have membrane bound organelles. So these are not found in bacteria, but they're found in all the other ones. Sometimes there's one really big one. Sometimes there's thousands of them, depending on the kind of cell If a cell needs a lot of energy, it's gonna need more mitochondria. So depending on activity level. So what kinds of cells in your body do you think would have a lot of mitochondria? Yeah, your muscle cells, because they're doing a lot of work. They need a lot of energy. So a muscle cell, if we looked at a muscle cell under a microscope that has the capability of seeing something like a mitochondria, you would see a lot of them. You'll notice here it has two. Wow, this is doing weird things today. So it has two membranes. It has two phospholipid bilayer membranes. It has an inner membrane and an outer membrane. So this is our inner membrane. And then this is our outer membrane. And in between those two membranes, there's a space called, it's really a great name, the intermembrane space. What's really important for us is not the necessarily intermembrane space, but the space within the inner membrane, which is the matrix. Are you too young for the Matrix movie references? Okay, that's all I ever think about when we talk about the matrix of the mitochondria is like we're in the matrix. I've never seen the movie, but I know that that's an important line of the movie, like being in the matrix. So we're in the matrix. So in the matrix of the mitochondria, there are enzymes. That are necessary for cellular respiration have to happen. Those are in the matrix, and they're also in these folds on the inner membrane. So they're both in the matrix and in the inner membrane self. These folds are called the cristae. And they're important because why would a folded membrane be better than a straight membrane? What does it provide more? More surface area. More surface area means more enzymes. More enzymes for cellular respiration means we can make more ATP. More ATP means more energy. So those folds are increasing the surface area of the inner membrane. So that means there's more enzymes there. So the enzymes are found both on the inner membrane and in the matrix. Also in the matrix, there are ribosomes. And there's actually DNA in the matrix of the mitochondria. And mitochondrial DNA, we're not going to talk about a huge amount of detail. But mitochondrial DNA is different than your other DNA in your cells. Mitochondrial DNA you inherit only from your mother. So it's called your maternal DNA. So if you ever have had a DNA test, you can get DNA tests, 23andMe, all those kinds of things. Sometimes they will do maternal DNA testing, and that maternal DNA testing is testing just your mitochondrial DNA, and you can trace your lineage back because your maternal DNA is exactly the same as your mother's maternal DNA, and your grandmother's maternal DNA, and your great grandmother's maternal DNA, et cetera, et cetera. So I did this like a long time ago through National Geographic, and they tested my maternal DNA, and I sent it to all my aunts and uncles on my mom's side because I was like, hey, we both got the same maternal DNA, and so it was just trace your ancestral lineage, and my ancestors are from Northern Europe, so nothing exciting with that. I was just hoping to find out something cool, but they came from Germany and the Netherlands. Anyway, that's mitochondrial DNA. Those genes in mitochondrial DNA will control enzymes and RNA in our RNA. But again, they're kind of cool because they're inherited only from your mom. Another organelle that is involved in changing energy from one form to another is a chloroplast. I feel like we need to change. Chloroplasts are famous for what reaction? Photosynthesis. So we all know that. So photo, the prefix photo means light, and synthesis means putting together. So this is the process of putting things together with light. So this is kind of the opposite of what the mitochondria are doing. Mitochondria are taking food, breaking down those bonds to produce energy. The chloroplasts are taking molecules using light energy to put them together. This is taking, so it's taking
solar energy and converting it to chemical bond energy. We're taking carbon dioxide and water, and we're making carbohydrates. And again, the point of this is that we're storing energy in those bonds between the carbons and the hydrogens and the oxygens. So we're changing the form of energy from solar energy to carbohydrates. And again, we'll talk a lot more about photosynthesis when we get to chapter 10. So in our next kind of unit, you'll notice there's some similarities here between a mitochondria and a chloroplast in terms of structure. They are also two membrane structures in that they have an inner and an outer membrane. So here it's somehow indicating inner and outer with these lines. This is the inner one. Here's the outer one. Just like the mitochondria, they also have DNA. And they have little ribosomes floating around. Ribosomes. There we go. Within the inner membrane, instead of the space being called the matrix, and it's not as cool in the chloroplast, it's called the stroma. So the stroma is that space inside the inner membrane. And you'll notice that inside the inner membrane here, we've got these little stacks of pancakes. Some people say they look like stacks of coins. These are called thylakoids, which are also membrane bound. So really it's kind of like they have three membranes. They've got the outer membrane, the inner membrane, and then the thylakoid membranes within the inner membrane. That's a Y. Thylakoids. And when you stack up a whole bunch of thylakoids, like here, that's called a granum. What do they do? So that this is where photosynthesis is happening. So the membranes of the thylakoids have the enzymes that are needed for photosynthesis. And also the pigments that give color. What's the main pigment found in chloroplasts? Chlorophyll. So when you get to this in lab, you'll actually take some plants and you'll separate out the pigments and you'll see that there's more than just chlorophyll and there's more than one kind of chlorophyll. But those pigments are also found there. What kinds of cells have chloroplasts? These are found in plants. And then there's also some photosynthetic protists, like algae. So not in animals yet. That would be a cool genetic engineering program. See if you can genetically engineer us to have chloroplasts. We can make our own carbohydrates. More reason to be outside, and you don't have to take time to eat. Chloroplasts are in a family of organelles called the plastids. So there's 3 examples that your book talks about. So chloroplast is one of them. A myeloplast is another one, which you are familiar with. Where did you see those in lab? What kind of thing had a myeloplast? What kind of thing did you look at? What kind of myeloplast? I can't know the better word than saying. I'm not doing very well with it.
You do?
Yeah, the potatoes. When you slice the potatoes, what did you stain it with? Iodine, and what happened? There was that purpley, dark purpley color because what's in the myeloplast? What's it storing? What made the iodine turn purpley? Starch. So myeloplasts are storing starch. So they're a specialized plastid that is there for storing starch. They're found in other tubers, but potatoes are the ones we talk about the most with that.
And a third kind of plastid are the chromoplasts. And these also store pigments. Chromo is color in things like fruits. So like orange and yellow pigments that are found in fruits are stored in chromoplasts. You don't think you saw them in the bell peppers? I think they just kind of look reddish. I don't think you specifically see the chromoplasts in that, unfortunately. Store pigments? Is that? Oh, second class is amyloplast. Okay, so those two organelles, mitochondria and chloroplasts, are changing energy from one form to another. Then we're kind of getting into the last bits that we don't put into a nice category easily. Peroxisomes are one of them. They don't really have a distinctive structure. Like this doesn't really look like anything specific when you look at it. And I think when you look at these cell models, you can look at these and say, okay, well, I know that's a mitochondria because it's got the whole inner membrane. And then there's these kind of just regular blobby things. Is it a vesicle? Is it a peroxisome? Is it a lysosome? I don't know. They all kind of look the same. But again, distinctive structures like mitochondria, chloroplasts, rough ER, smooth ER, Golgi bodies. These are chloroplasts that are not cut in half. Those are things that you should recognize, but again, these don't have any kind of distinctive feature that makes them stand out. They're important because they have an important jobs, but they don't look particularly spectacular under the microscope. These, just so you know, are chloroplasts. So these are the stacks, these would be the granum. And this is a mitochondria, which doesn't look as distinctive, but you can kind of see some of the inner membrane folds in there. So the peroxisome, again, doesn't look like anything special. It only has one membrane. So it's not a double membrane bound organelle, but it's still membrane bound. And it has enzymes in it that take hydrogen molecules from hydrogen atoms from molecules and they add it to water. Sorry, to add to oxygen to make water and peroxide. So really they make peroxide first. That's why they're called peroxisomes. So they're taking hydrogen ions off things that might be toxic to the cell and putting them in peroxide, but peroxide also is not great for the cell. So then they have further enzymes that will break this down into water and oxygen. So they're detoxifying stuff in a kind of multi-step process. You've seen the peroxide reaction. I'm sure at some point you've had a cut and you put peroxide on it and it bubbles. Those bubbles are the oxygen and it's changing into water and killing stuff as it does it because peroxide is toxic. They also can break down fatty acids. So again, they have enzymes to break down fatty acids and they can detoxify alcohols. So they're kind of like lysosomes and they're metabolic compartments. They have enzymes to help metabolize things. And again, no distinctive structure necessarily.
What makes a peroxisome and lysosome look different?
The kinds of enzymes that are in it. So like a lysosome would have enzymes that function at acidic pHs, so it maintains an acidic pH, and it's going to break down things like proteins and carbohydrates. This is more fats and alcohols and that kind of thing. So it's just the enzymes that are in it that will process different kinds of molecules. But they don't really look any different. Okay, questions about any of that? We've kind of finished, oops, with organelles. I thought there was, oh, wait, 1 second. So let's take a minute and make another Venn diagram because why not?
So, thinking about plant cells and animal cells, you can't see it.
Take a minute and think about differences in organelles that are present. In plant cells versus animal cells. And then what is present in both? Okay, wait. I'm realizing why I kept it later. Let's come back to this. Make yourself a Venn diagram. You can think about it. We got to talk about cytoskeleton first. Spoiler alert. One of the answers is in the cytoskeleton. Okay. Sorry.
Cytoskeleton. Cyto means
Cell. Skeleton is like skeleton. So this is the cell of the skeleton. The cell of the skeleton has different kinds of bones, just like you have different kinds of bones in your body. And you can see some of these in this picture. This is a picture of a cell that has been tagged fluorescently. So you can see there's different colors in here. So you've got this orangish color, this green color, and this blue color, which is not actually the cytoskeleton, but this green is showing you microtubules, and the reddish orange is showing you microfilaments. The intermediate filaments aren't on here, and this is just DNA. This blue is DNA. They have different compositions, so we can tag them with different fluorescent molecules and then look at them under a fancy microscope that shows us where they are found in the cell. So again, green is your microtubules and orange is your microfilaments. So what would you think the job of the jobs of the cytoskeleton are? Structure, so giving support to the cell, helping it with it maintain its shape. So we think of cells as being tiny, and you probably don't think of them as needing support and maintaining their shape, but that is important even on a cellular level. This is especially important for cells that don't have a cell wall, like animal cells that don't have a cell wall. So this is providing internal support instead of that external cell wall support. That plant cells still do have cytoskeleton, but not in the same way. So those are ones that you might think about off the bat. What else? There's two other things that are important. Things that your skeleton helps you do too, that we don't always think about in conjunction with other things sometimes. Movement. So you probably don't think of your cells as moving around a lot. And it's not necessarily your whole cell, but within the cell. So it can help with movement within the cell, but also the cell itself. Your cells need to be able to move, and they need to be able to move things around within the cell. And so the cytoskeleton provides that framework for that to be happening. And then we talked about last class a little bit, the process of phagocytosis, but being able to move cell membranes to shape the cell for certain processes. So phagocytosis is when a cell sees something it wants and it brings it in. So being able to move and manipulate the membrane to change the shell, the shape of the cell to be able to bring something in. That's facilitated by the cytoskeleton. So supporting it, giving it shape, maintaining the shape, but also changing the shape, and then moving things within or moving the actual cell itself. So there's three different kinds of fibers that are involved in the skeleton. So this is the first one. Microtubules, which we saw on the previous slide, were in green, and you'll see they're in green here also. So these cells, we can see the tubules as well. Those look like some kind of epithelial cell, so like skin cell or something like that. Microtubules are hollow tubes. So they are not solid. You can see here, they're hollow, and they're made up of the protein tubulin. Tubules are made from tubulin. That should be a little easier to remember than some of the other things you have to remember. They're what we call a dimer, meaning they have two different forms of tubulin. So you can see here, we have alpha tubulin and beta tubulin, and they're forming this helixy shape, like an alpha helix, secondary structure of the proteins, that's going to create that hollow tube. These are found in all eukaryotic cells. So plants, animals, fungus, protists, all of them have microtubules. But they have different jobs in different kinds of cells. So they're, oops, not yet. They help maintain shape. They resist compression. So if something is trying to mush the cell, they're gonna push back against that. They're kind of like the girders in a building or the rebar in a concrete structure. So they're compression resisting as they maintain that shape. They're also involved in cell motility. And in this case, we're talking about cilia and flagella, which you weren't supposed to read about all the details of that in the textbook, but there's lots of details if you want it. These are showing you cells in the trachea, and these are all little cilia, all this top layer up here. These are all cilia. So you have cilia on your trachea cells that are helping to move particles out of your respiratory system. So if anything's getting into your lung system, those cilia, also microvilli in the intestines. Whoops, sorry, microvilli in the intestines is the next one. Sorry, I was like, that doesn't look like that. And then this is a sperm. Sperm has one long flagella that's made of microtubules. It's also, these are responsible for chromosome movement. When we're talking about cell division, so when cells divide, chromosomes have to move all over the cell to get to the right place at the right time. And that movement is controlled by the microtubules. This is showing you here, this is what we call a centrosome. They're found in animal cells, and they're where the microtubules are connecting and the actin fibers are connecting to be able to move. They're like the anchors for moving chromosomes. I think they look like pasta every time I look at them in the cell model. They look like ziti to me. I don't know if I'm just hungry all the time, but that's how I remember them. They're like these little ziti tubules. And again, those are only found in animal cells, and we don't know why plant cells don't have them. We don't fully understand how they move their chromosomes without that, but animal cells have those. They also can move organelles. So you can see here, here's a vesicle, and this vesicle has like little legs. Okay, it's being moved. This is called a motor protein, sorry, not proton. This is excellent. And this is a microtubule. So this is a process that's powered by ATP. And this vesicle can be like moved by a motor proton that is actually like walking along the microtubule. And I thought I had a video of it and it didn't work. So I got to find a video. I'll post a video on Moodle. There are more of them. Okay, this is what your life is when you become a science person. You watch videos of motor protons carrying vesicles around the cell and you think it's cute. Because they're cute, they're talking, they're like little living things inside your cell, and that's happening, and it's crazy. I delight in the details of this sometimes. Okay, a second kind of cytoskeleton, of bone that you might find in a cell are the actin filaments or the microfilaments. So these are solid fibers. They're not hollow tubules, they're solid rods. So tubules is kind of telling you it's a tube. These are solid rods. They're made up of two actin strands that are wrapped around each other. So you can see it just looks like if you zoom in on thread, it looks like just twisted thread. Actin is just a protein. So it's just actin proteins that are twisted around each other. So these are also important for cell shape, but they're not compression resisting, they're tension bearing. So they're able to support tension. They also help with changing cell shape. And the classic example of this is the amoeba, which you see down here. So amoebas, you didn't look at amoebas yesterday, right? I mean, this week in lab. Did you look at amoebas? I think it might not be till next semester. Amoebas have the ability to just change their whole body shape all the time and make these little projections of cytoplasm that are called pseudopodia, which is like false. Pseudo is false, pod feet. So they just move by changing the shape of their cell membrane. So moving the cell membrane and the cytoplasm following will change the way that they are shaped and the way that's how they move. This is showing you the microvilli in the small intestines. And these are supported by, so again, tension bearing, they're supported by the microfilaments. They also, because they're cell shape, thinking about cell shape, an important cell shape change that happens in the body is muscle contraction. That's an N. So in muscle cells, and then also in which, did you see this in lab? Did it work? When you looked at the Elodia under the microscope, if you left it there for a while, did you do this? No. Yes, some of you did, some of you didn't. If you leave Elodia under the microscope for a while, they will actually start to concentrate their chloroplast near the cell wall, and they will actually start to move around, moved by microfilaments. And they're also important in the process of dividing for animal cells. So different structure and some different function because of that structure. And then the third one are called intermediate filaments. So where do you think they fall in terms of size? In the middle. Okay, so microfilaments are the smallest, microtubules are the biggest, and intermediate filaments are in the middle. They're made of, they're made of different fibrous proteins. So actin and tubulin are globular proteins. These are fibrous proteins, and they're found in the keratin family. There's different ones, but they're all in the keratin family. Keratin is the protein that makes up your fingernails. But they're made up of several different ones. I should have gone down there, sorry. They're woven together, interwoven into cables. That's a cable shape. So giving it strength. They also help with cell shape in that they are tension bearing, just like microfilaments. They help to anchor the nucleus and other organelles. So keeping them where they need to be when they need to be there. And way back when, like on Monday. I'm sorry, I'm talking and writing. We talked about how the nucleus had like a structure on the inside of it called the lamina on the inside that helped to give support. That lamina is made up of intermediate filaments. The other filaments, the microtubules and the microfilaments, will often, if they're needed somewhere else, they can be disassembled and moved somewhere else and reassembled where they're needed in the cell. These are a little bit more permanent. The intermediate filaments don't move around as much. So like the outer layer of your skin is made up of mostly dead skin cells that are supported by the intermediate filaments, but they're not moving all around all the time. Okay, now we've talked about enough. See what you can do with this. What do plants have? What do animals have?
Okay, let's talk about this.
What do plants have that animals don't have? Cell wall, which we didn't even really talk about. We're gonna talk about that in a minute. What else? Chloroplasts. Anything else anyone thought of? Central vacuole. So remember, we talked about that last time. That's the really big open space that has a lot of water and some other ions and stuff like that. How about animal cells? What do animal cells have that plant cells don't? So lysosomes get weird because some people say that plant cells don't have lysosomes, and some people say the peroxisomes are kind of lysosomes, so they do, but they're different. So I still kind of put them in the middle because it depends on what camp you fall in. But you're right, there is some people that would say, yes, animal cells only have lysosomes. There's really only one thing that I have from your book that animal cells have that plant cells don't have. That's the ZD, the centrosomes. So that structure of microtubules that is used for cell division. Okay, what do they both have? Lots of stuff. Go. Cell membrane. More. Nucleus, yes. Mitochondria. Ribosomes. What? Microtubules. I'm just going to be lazy and put cytoskeleton. Even though, okay, this is another thing you can put under animal cells. Only animal cells have intermediate filaments, not all animal cells, but they're only found in animal cells. But cytoskeleton, microtubules, and microfilaments are both found in both, but more so in animals because plants have the cell wall. What else? What other parts of the endomembrane system that we talked about last time? Endoplasmic reticulum, both rough and smooth. The Golgi body, or the Golgi apparatus, whichever one you like to call it. And lysosomes we're kind of putting in there. And peroxisomes. Am I forgetting anything else? I think we're good. You can think about nucleus, other structures within the nucleus, like nucleolus. You could put that in there too. Okay, we're gonna get done. We're gonna do it. Okay, we did not really go over the plant cell picture. You did it for your homework, I know. We talked about chloroplast, but we didn't look at it in the picture. This is the central vacuole. That's the one that's holding water. That helps to hold cell shape. This outside layer, which we didn't really talk about, is the cell wall. The cell wall really is an extracellular structure. It's outside the cell. It's providing protection for the cell. It's helping to maintain the shape. And it prevents too much water from coming in. Okay, you're gonna talk about in a couple weeks, maybe
next week you have a lab exam, but the week after. You're gonna talk about diffusion and osmosis, and we're gonna talk about it in lecture next week. Your cells, when they get too much water and can explode, plant cells can't, because that cell wall prevents that from happening. What's the cell wall made of? Which carbohydrates that plants have that we don't really use? Cellulose. So it's made up of cellulose. There's also some proteins and other polysaccharides in there, but cellulose is the primary component of the cell wall. And cells can actually have more than one. So as a plant cell is developing, it will grow a primary cell wall. And what can happen is it either turns into a thicker cell wall, a thicker primary cell wall, as the cell develops and matures, or depending on the kind of cell, it may actually develop a secondary cell wall. And that happens in wood. That's why trees are so strong. They've got two cell walls supporting their cells. So again, this is present in plant cells. What other kind of cell has a cell wall? All the way back to Monday. Bacteria, yeah, prokaryotic cells have a cell wall also. Different composition, but same idea. Okay, we may not get through all of it, but we're gonna get through this part at least. So if you look at this cell wall, and if you looked at the cell models in lab, and you looked at a slide, a picture at least, they have these holes in them. Because cells can't just live in a multicellular organism. Cells can't live on their own. They have to be able to communicate and interact with the cells around them. And so plant cells communicate and interact. They're connected to other cells through plasmodesmata. So these are a kind of cell junction. Between the plant cells, they're literally channels. And between the cells, the membrane can be continuous. Meaning the plant cell, this plant cell's membrane is joined to this plant cell's membrane, because this is a whole plant cell too. And so their environment between the two cells is exactly the same because they're sharing everything. It's like a covalent bond between cells. So the membrane is continuous, which means the environment is the same inside both cells or all the cells that are connected by the plasmodesmata. So that allows plants, large multicellular plants, to be able to share materials easily between different cells because they can pass through those plasmodesmata openings, those channels in the cell wall. That is where we're going to stop because there's no way we're going to complete animal cell production control. If you did not complete that, sorry, I didn't bring the first page up. Don't forget the twins. Homework is due Monday.
This was in the seat.
Oh, dear.
My seat.
Thank you. Yes. We will hunt down somebody. There's a driver's license. Thank you.